Welcome to people as you join. We'll just allow attendees to join before starting. Okay, let's make a start. Good morning, good evening, wherever you are. My name is John Draper, and I'm the Vice President of Research and Training at the Canadian Stem Cell Network. Just do a little bit of an acknowledgement here. So while we meet today on a virtual platform, I would like to begin by acknowledging the Indigenous peoples of all of the lands that we are on today. The work of Canada's Stem Cell Network occurs from coast to coast and occurs on the traditional, ancestral and unceded territory of many of the Indigenous peoples and nations, including the Anishinaabe, Algonquin Nation, where SCN is headquartered. So Stem Cell Network honors, recognizes and respects these nations as the traditional stewards of the lands and waters where we work and meet. With that, I would like to start by saying I'm very pleased to welcome everyone to our first session of the Global Regenerative Medicine Trainee Speaker Series, hosted in partnership with the Australasian Society for Stem Cell Research, the German Stem Cell Network, the Japanese Society of Regenerative Medicine, the Belgian Society for Stem Cell Research, and the Korean Society for Stem Cell Research. We would like to thank a dedicated committee of trainees drawn from each country for helping to organize these sessions and selected speakers. The names of those on the training organized committee are shown here right now. So we're very excited to launch this seminar series, which will profile cutting-edge research advances from the next generation of stem cell leaders across the world. It is a really exciting opportunity to see what's happening here in regenerative medicine and stem cell research globally. The sessions of this seminar series are currently scheduled to be held every 2 months until January. So if you're interested in attending future sessions, please check out the Canadian Stem Cell Network website to see more details and register. We will drop the link in the chat, which should happen soon. I also encourage everyone to check out the training section of Stem Cell Network's website, and also your home country's network or society webpage to learn more about other training opportunities or knowledge mobilization opportunities like this. I just have a few housekeeping notes before I pass over to our trainee committee members to facilitate the session. The session will be recorded and posted publicly on Stem Cell Network's YouTube channel. Each speaker will have up to 15 minutes to present and then a few minutes for questions. We really do encourage you to put your questions in the Q&A chat box located at the bottom of the screen. It's vital that you actually engage with this. It's vital for the trainees to get the questions they deserve for the hard work that they've been performing to bring you these results, exciting insights that they've been doing. And it's really helpful for you to actually engage in this process. So please do. Closed captioning is enabled for the session, so if you need that, please use that. You can choose to show or hide this in the options at the bottom. And with that, I'll hand it over to our first trainee committee representative, who is from the Canadian Stem Cell Network, to introduce our first speaker. Lauren, over to you.
Thanks, John. My name is Lauren Durland, and I am based in Canada. Since this is an international series, we'd love to kind of find out where all of you guys are joining from. So feel free to post what country you're joining from just in the chat below. I am delighted to welcome the first speaker on behalf of the Canadian Stem Cell Network. The speaker for this session is Alexander Baker. Alex is currently a postdoctoral fellow at the University of Toronto in Canada and will be speaking about his work on how hydrogels emulate the extracellular matrix for unbiased in vivo and in vitro organoid formation and drug testing. Alexander, please go ahead and begin your presentation whenever you're ready.
Thank you very much, Lauren, for the kind introduction. For several decades, researchers have focused on trying to recapitulate the natural morphology present in many tissues in the endocrine system. One of these architectures is the branching networks found in the formed by mammary epithelial cells. These are composed of ducts that are primarily containing luminal and myoepithelial cells. And one of the challenges that has largely been solved looking at pathway signaling is the importance of the extracellular matrix, notably, as shown in this figure, collagen and the basement membrane that can contain things like laminins and fibrinogen that give rise to the biochemical signaling required for these complex tissue architectures to form. Furthermore, the importance of proliferative signaling and cell death signaling gives rise to these hollow architectures seen in tissues. But so far, it's been quite challenging to be able to recapitulate these in vitro to the extent that is found in our native tissues. And to make this matter even more challenging is that in the in vivo context, we have a very dynamic environment, especially in the cases of diseases like cancer. So one of the examples is that these typically hollow myoepithelial ducts and luminal ducts can become filled with cancer cells as they proliferate rapidly. And over the course of the disease progression, an increase in tissue stiffness has been characterized due to the increase and changes in the extracellular matrix, notably things like collagens and the stroma, increases and changes in levels of hyaluronan. So hyaluronan is a material that is near and dear to me. It's one of the materials that I used extensively during my PhD. And the reason that hyaluronan is such a useful material for chemical engineers and biologists is because we can chemically modify hyaluronan to generate hydrogels. And that's important because working with hyaluronan in vitro is extremely challenging when it comes to mimicking the properties of the hyaluronan that exists in an in vivo context. Specifically, natural hyaluronan is a very high molecular weight. Meanwhile, the molecular weights that are available for researchers tends to be quite low. So in order to form these materials that are useful for in vitro cell culture and tissue models, disease models, we use chemistry. And in particular, the chemistry that I'll be talking about a bit today is oxime chemistry, which is the formation of a cross-linked product in the presence of an aldehyde or a ketone. So hyaluronan hydrogels are quite tunable for 3D cell culture. And this is important because in many tissues, we have a range of mechanical environments that cells see. As a control, we compare our hydrogels to matrigel, which is a protein extract from the Engelbrecht swarm mouse sarcoma. This is composed of a mixture of proteins found in the extracellular matrix, but importantly, lack hyaluronan, which we know is crucial and present in almost every tissue in the body. And you can see from this data in the gray bars that we can tune the stiffness of these hydrogels by altering the ratio of the modified polymers. And in red, we show that the stiffness of these materials mimic quite closely to what's found in a mouse tumor, which is important because often drug screening is done in these in vivo models. These materials are completely compatible with encapsulating cells, and they're well distributed in a 3D given the rapid gelation rate, typically less than 15 minutes. And this image on the bottom is showing that these cells are capable of proliferating to form multicellular tumor spheroids over the course of several days and weeks. And the longest that I've grown these cells in hydrogels is more than 4 weeks. So with this hydrogel, and this was work that I had finished during my PhD, and then I'll talk about some of the new challenges we've been able to solve. But one of the existing challenges was the fact that there are many materials that are published in the literature, but very little was known about exactly how well they faithfully recapitulate the disease. So to solve this, we looked at signaling pathways that are canonical in breast cancer specifically. So these pathways cover things involving cell fate, cell survival, and genome maintenance. And we benchmarked the gene expression levels of breast cancer cells that were cultured either in our 3D dimensional hyaluronan hydrogels, in matrigel, or on 2D tissue polystyrene. And we compared that to tumors grown in orthotopic mouse xenografts. And when we look at the pathways that were differentially expressed in these in vitro models, what we noticed was hyaluronan actually maintained these pathways similar to the xenograft model, whereby matrigel and 2D dimensional culture actually perturbed these pathways, which was a bit of a red flag, so to say, in terms of being able to trust the in vitro data from those two-dimensional or matrigel models, where hyaluronan, we know, plays such an important role in breast cancer and did seem to keep these cells expressing those pathways as expected. But one of the challenges of working with that hydrogel that I discussed was that it was not very dynamic, meaning we used hyaluronan, which is important for the biochemical signaling. But when it came to the ability for cells to remodel it, the polymer crosslinks were done using polyethylene glycol, which is a very inert biopolymer. So in this work, what we did is we used a dynamic peptide crosslinker, which we denote as MMP-OA, which means it's an oxyamine modified matrix metalloprotease peptide, which is cleavable by MMPs 2, 7, and 9. And this material was quite stable in vitro over the course of 4 weeks, as I show. But when we add either hyaluronidase or collagenase, we see that these materials are capable of being degraded, which is important because we expect that all of the cells we would encapsulate will have the ability to remodel using either of these mechanisms. And in this work, we explored polarization of breast mammary epithelial cells, the ability to expand tumor cells, and forming patient-derived xenografts. So just as I showed with that other hydrogel, the one that was cross-linked with polyethylene glycol, when we cross-link this hydrogel with the matrix metalloprotease cleavable peptide, we are able to still form a tunable material in a range that is similar to the native tissue. And importantly, when we incorporate extracellular matrix proteins, collagen 1 or laminin, we have a material that has a similar mechanical property to matrigel. One of the things that really surprised us was that the inclusion of these proteins actually changed the mechanical properties beyond the bulk storage modulus. We found that adding these proteins actually introduced stress relaxation properties. And the best way to think about that is when you're pushing on something like silly putty, it doesn't just break, it relaxes and is able to move. And that's important because in our bodies, the extracellular matrix isn't formed of a significant amount of covalent bonds. It's largely physical interactions where the cells can push through the matrix. And as you can see in this green line and the blue line, we have similar stress relaxation properties. So the next question was, could we work at something other than the cancer cells? And one of the examples of an immortalized healthy epithelial cell are the MCF10A. And this was efforts that were started even before I started my PhD, but I had long kept in the back of my head as a problem that I was hoping to solve. And I was pleasantly surprised to see that introducing laminin that gave rise to these stress relaxing properties allows us to form these acinary structures where you can see the nuclei in blue and a hollowed out structure. In collagen, which is typically present when there's fibrosis occurring or in diseased tissue, we see that this architecture is actually lost. And of course, in matrigel, we have the expected morphology. When we characterized the number of spheroids, we saw that collagen had reduced spheroid number compared to the other two scaffolds. We characterized the gene expression levels of several pathways, and we were really interested to see in particularly by having CD44, which is present on all cells with the exception of a few, which is actually known to bind to hyaluronan. The presence of hyaluronan in our materials shown in the green and red gave rise to a decrease in expression of EGFR compared to matrigel. So this is showing that these MCF10As are actually interacting with the matrix and not just the presence of laminin or collagen alone. We also characterized other genes, but they were all conserved between the HA-oxime and matrigel. And these genes represent extracellular matrix or mechanical signaling pathways. One of the important aspects of these cells forming these hollow acinary structures is that they actually secrete their own extracellular matrix once they're implanted in a compatible environment. And if that material allows that material to be sequestered and captured, as we can see, this laminin 5 was indeed expressed by these cells and is contained on the periphery of these acini. Beta-catenin is a cell-cell junction marker, and we see evidence of beta-catenin, which mimic that found in matrigel. We wanted to understand the mechanism of the beta-1 knockdown, and we wanted to understand the mechanism of why these cells were forming these acinary structures. So we performed an siRNA knockdown targeting beta-1, which is one of the integrins that interacts with laminin-1. In this knockdown, I'm just showing several examples of where you'd have an acini and a non-acinary structure formed. So even in the case where you have untreated cells, the formation of acini is not guaranteed, and this is the same thing in Matrigel. And it's quite common amongst researchers to have to screen numerous batches of Matrigel to be able to get this very desired phenotype for studying healthy tissue. We found that the knockdown of integrin beta-1 resulted in a decreased number of polarized spheroids, as shown in the red, compared to the other three groups, which was exactly as shown with Matrigel. And we also saw that the number of spheroids was reduced when this beta-1 integrin was knocked down. And this data was analyzed double-blind to make sure that there was no bias. We also characterized the beta-1 expression at the protein level using flow cytometry, and I'd like to credit Amber for her work doing this. And in this case, we can see that the beta-1 integrin expression is decreased for up to 3 days, while based on PCR methods, we saw even at day 6 that there was a decrease in the amount of mRNA that was used to express the beta-1 integrin. So being able to show that this worked with healthy cells was quite important, and this gave us confidence to go forward looking at a variety of other tissue types. And that's because, as I mentioned, hyaluron is expressed in almost all tissues. So we explored a variety of cells from other tissues, such as blood, liver, brain, breast, and lung. And in this case, the immunoimages are showing CD44 as well as nuclei. And you can see the broad diversity of morphologies present in these cells. We also looked at pancreas, colon, and prostate cells. And again, we see that these cells survive and proliferate within these materials. In the one instance where we have a CD44 negative cell line, these are the ovarian cancer cells, they do not express CD44. But this does support the fact that this material could actually be useful for many other tissue types beyond looking at the breast epithelial cells. So then in collaboration with Dave Sescon, we had access to primary patient cells. So these were cells that were taken from a patient's biopsy and cultured directly into our hydrogel. We see that all of the hyaluronan hydrogels facilitated the growth of these very valuable and sensitive cancer cells, similar to Matrigel, which is the standard for how the Sescon lab and other labs in the world expand primary and patient cells. Importantly, these cells do proliferate beyond the images that I show on the left, showing over the course of 14 days, we do have an expansion of cells, albeit quite slow compared to some aggressive cell lines grown in 2 dimensions. We also performed drug screening. So one thing I would like to highlight is something called the growth rate metric. And this is used to assess the fact that when cells are grown in vitro, they grow at different rates. So this is to take into account the fact that the drug will impact growth rate, as opposed to just looking at the number of live and dead cells. This will help identify drugs that are cytostatic, meaning they prevent cells from proliferating, as well as cells that actually cause cell death. So these first 3 drugs are all drugs that are used. Okay, thank you for the warning, Lauren. All of these drugs are used as cytostatic drugs, meaning they arrest cell growth. And we see largely similar performance across these materials. And we also looked at 2 other drugs that are known to target the RAC-GTPase and PI3K beta. And these were pathways that we highlighted in our previous work as ones that would be causing differential drug response compared to Matrigel. And we see that that response holds true. Then we looked at being able to use these materials as a means to deliver these patient cells in form of patient-derived xenograft. We established patient-derived xenografts using 3 different hyaluronan hydrogels and compared them to Matrigel. And we were surprised pleasantly to see that our hyaluronan hydrogel outperformed Matrigel in terms of being able to successfully generate patient-derived xenografts. And one thing to note is the extremely long timeline required to generate patient-derived xenografts. This is an important thing to keep in mind if we're going to use these as tools for drug screening. When it came to characterizing these tumors, we looked at the immune cell composition, and I just want to highlight that the population that we noticed the biggest difference was with regards to the monocytes. And when we look at the macrophage population, we see that Matrigel causes a bias of the M1, which is the immune responsive, and M2, which is the more like the healthy state immune cells, in terms of how the tumor cells, how the host immune cells respond to the materials. So I want to thank our collaborators Dave Sescon and Jen Cruikshank, as well as Laura Ballman, who's now a postdoc at MIT. And my lab is now started at Dalhousie University, and I thank you. I'll take any of your questions.
Thank you, Alexander, for a fantastic talk. This was a really nice overview of your work, and I think we're all so excited to see what comes next. We now have time to take a few questions from the audience. So if you'd like to ask a question, please just put it in the Q&A chat box, and I'll read the questions out loud to our speaker. Okay. We have a question from Ritika, who's one of our panelists. Ritika says, beautiful talk, Alex. In the different cell types you have tried, do you know how primary cells compared to iPSC-derived cells of the same type? So for example, primary endothelial cells versus iPSC-derived endothelial cells?
That's a very interesting question. We haven't looked into extensively using iPSCs. So majority of the cells we've looked at have been either established cell lines, or in the case of the breast cancer cells, those were primary patient cells. So I think this opens more questions for where else these materials can be used, to looking at endothelial cells, for instance, or even neuronal cells.
Cool. Now we have some audience questions. Our first one, do you think that these hydrogels would support human pluripotent stem cells? I guess that's kind of a similar, but I guess for that cell type specifically, what do you think?
Yeah, so there was actually some work early on looking at astrocytes and iPSCs to be differentiated towards neuronal cells, and the material tolerated those cell types quite well. I think the mechanical properties being similar to Matrigel and having things like laminin really make it a very compatible environment for many of those cells, which require typically coated wells or coated plates to facilitate cell attachment and growth. So I certainly think that would be possible.
Cool. We have another question from Farishte. She says, this is a great talk. Did you see DAPI intensity with hydrogel with collagen or hydrogel plus laminin, as DAPI can show chromatin condensation, which is defined by stiffness?
That's a really interesting question. That's something I didn't even really come across. So in a lot of the work where I do nuclear staining, I used either host or DAPI. In the case of DAPI, I don't tend to do very high magnification images with these three-dimensional hydrogels, because it's always a challenge to get a focal plane that you can see many of the spheroids. So I think that's an interesting question for looking at DNA structure within the cells, but that kind of went beyond what our focus was.
Cool. We have a couple more questions. I think we probably have time because they look pretty quick. The first one is, how feasible would these materials be for other labs to introduce globally? Yeah,
that's a great question. So interestingly, when we first started this work, it began because of a collaboration with an industry partner. And for the sake of everything, I'm not going to mention who that industry partner was, but the material was exchanged to that lab, and they were able to use it to grow cells in vitro. The synthesis methods have been published, and numerous other graduate students in the Scheuchert lab have gone in and used this material as well. So I think that does act as a testament that is translatable for other researchers to use. There is a bit of hands-on, getting used to it technique that always happens with science.
Cool, it's super interesting. And this will be, I think, our last question. Do you think we need to use different hydrogel mixtures for different cell types? Is this something that you think could be standardized to different cell types, or would you have to make slight adjustments to support different cell types?
Yeah, I think all of us would like it if we could just use one hydrogel for every cell type, right? It would make it a lot easier in the lab. But I think the reality is, based on other work that has gone on in the field, we noticed that certain phenotypes require certain things, whether it's porosity, different extracellular matrix components, density of those components, and mechanical properties as well. So I think the state is still, you have to check before you really study a certain tissue type or disease to really know. But I think based on this work, we certainly saw evidence that it was at least compatible for a variety of tissues, similar to Matrigel. So perhaps it is possible, but I wouldn't say absolutely.
Awesome. Thanks again, Alexander, so much for taking the time today to share with us a little bit about your work. We encourage audience members that might have further questions to contact Alexander and keep the conversation going. I'll now hand it over to Yoshitaka, who will introduce our next speaker.
Thank you, Lauren. My name is Yoshitaka Kase, and I'm happy to welcome the next speaker on behalf of the Japanese Society for Regenerative Medicine. The second speaker for this session is Yosuke Yoneyama. Dr. Yoneyama is currently an assistant professor at the Tokyo Medical and Dental University, and will be speaking about their work on modeling human fatty liver disease with iPSC-derived organoids. Yosuke, please go ahead and begin your presentation.
Okay, thank you very much for kind introduction. Do you see my slides?
Yes, okay, go ahead.
Okay. So first of all, I'd like to thank organizers of this seminar and Stem Cell Network, and also appreciate JSRM to give me this opportunity. Today, I'm going to talk about human iPSC-derived liver organoid to study fatty liver disease. Before starting, I thank Takanori Takebe and his lab members in my university, as well as in Cincinnati Children's, for supporting this work. So non-alcoholic fatty liver disease, or NAFLD, is strongly associated with metabolic disorders such as obesity and insulin resistance. This disease ranges from fatty liver or steatosis to the progressive steatohepatitis form, or NASH, which leads to inflammation and fibrosis. Such pathogenesis is driven by multiple cell types in liver, including hepatocytes, as well as immune cell types like macrophages and fibrotic components called hepatic stem cells. Since NASH is the leading cause for chronic liver failure, early detection and treatment are crucial. However, there are no effective drugs available. So one of the reasons behind this is that NASH pathogenesis is highly heterogeneous among patients. So there are critical needs to better understand the traits of vulnerable populations and develop preventive and therapeutic strategies. Recent studies have shown that there are several common genetic variants which modulate the risk of NAFLD as well as the severity of this disease. So for example, these variants include PNPLA3, TM6SS2, MBOT7, and GCKR, which increase the risk of the NAFLD. And HSD70B13 and mTORC1 have been recently implicated in the protection against NAFLD. However, the contribution of these genetic variants for clinical phenotypes remain to be established because numerous non-genetic confounders like lifestyle and nutrition are varied among patients. In addition, when the genetic effects are intersected with the metabolic trait, it is further difficult to interpret the genetic variants in the pathogenesis. So in my lab, to tackle with these issues, we decided to take advantage of human iPSC-derived organoid model that allows us to exclude environmental factors under defined culture condition. In this study, we aimed in the dish genotype-phenotype association approach using human liver organoids derived from multiple distinct genotyped donors. To achieve this concept, there are two challenges. First, we have to generate liver organoid that contains known hepatocyte lineages, essential for modeling inflammation and fibrosis as seen in NASH conditions. Second, we need an efficient platform to screen multiple donor organoids for studying the genotype-phenotype associations. For the last few years, our lab has been developing the protocol for generating human iPSC-derived liver organoid. We applied a directed differentiation approach starting from definitive endoderm, but we also produced mesodermal lineages in parallel, resulting in the generation of diverse cell type in the culture. This human liver organoid, or HLO, have nicely coordinated structure shown this, where stromal and immune cells are surrounding the hepatocytes. The cellular diversity was confirmed by single-cell RNA sequence analysis, showing the presence of zonal hepatocytes, as well as other components, including stellate cells and Kupffer cells. In addition, we validated the physical functions of HLO shown here. Next, having those organoids, we found that the supplementation with a free fatty acid, oleic acid over time, causes massive lipid accumulation inside the organoid. In this organoid, we call them steatohepatitis HLO or SHLO. SHLO lost the ability to respond to insulin, looking at the insulin-stimulated suppression of hepatic gluconeogenesis, shown here. Furthermore, we recognize the increased production of inflammatory cytokines in SHLO culture. Eventually, SHLO develops fibrotic phenotype confirmed by immunostaining of extracellular deposition of type 1 collagen. So these results indicate that our HLO model can recapitulate a key aspects, including hepatic insulin resistance and important traits of NASH pathogenesis in a dish. Next, we try to devise a population-scale platform that enables us to study genotype-phenotype association using liver organoids. First, 24 donor iPSCs are directly differentiated into foregut cells. Foregut cells are preservable and expandable. So these foregut cells are embedded into a single dome of matrigel and specified into HLO. After maturation, this culture contains 24 different donor-derived HLO and allow for unmasked phenotyping. Then they are exposed to oleic acid and insulin-resistant steatohepatitis-like condition is induced. Finally, steatosis levels are live imaged at a single organelle level, followed by donor decoding according to the donor-specific combination of genetic variants in gene sequencing, and then key genetic variants associated with steatohepatitis are inferred. So this is the result of our screening. In the left panel, we had genotype profiling data from each donor HLO, including known NAFLD-associated genetic variants. And in the right panel, we got a quantification of lipid accumulation in each donor HLO, followed by ranking them from higher to lower steatotic phenotypes. Then we calculated the odds ratio of each variant, and as a result, PNPLA3 and GCKR, which are known variants for human NAFLD, were statistically significant in our screening. Our particular attention is GCKR because this gene is one of the most pleiotropic variants in human genome. GCKR is a glucokinase regulatory protein, which is playing a role in glycolysis pathway. Upon binding to glucokinase or GCK, GCKR inhibits GCK activity and then decreases glucose utility. This is the story of wild-type variant. In contrast, the NAFLD risk variant TT disrupt the binding to GCK, resulting in the increased glucose utility. So to assess the pathogenesis of these variants, we selected 3 donor iPS lines comparing to non-risk form CC and risk form TT and gene-edited TT iPSC converted from CC genotype, and then tested the causative impact in an isogenic background. The results we obtained are very clear. Compared with GCKR CC organoid, GCKR-TTHLOs accumulated more lipids in body peak staining shown here. And as expected, GCKR-TTHLOs shows higher GCK activity, which eventually leads to enhanced de novo lipogenesis. We also found that in SHLO carrying GCKR-TT variant, mitochondrial dysfunction is a key driver worsening the steatohepatitis phenotype, including inflammation. In addition, we analyzed the clinical cohorts by subdividing NAFLD patients into diabetic or non-diabetic groups. What we found here is that GCKR-TT variant elevates disease severity only under diabetic state, and it rather protects from NAFLD under non-diabetic state. From this result, we propose a mechanism of GCKR-TT variant explaining how metabolic associated genetic traits appear in human NASH. As a genetic effect, GCKR variant enhances glycolysis and lipogenesis, which together generate oxidative stress to mitochondria. At the same time, the presence of diabetic condition as a metabolic effect leads to hepatic insulin resistance that also worsens the mitochondrial dysfunction. Eventually, these two effects triggers vicious NASH cycle. We are also interested in another variant identified in our screening named PNPRN3. In contrast to GCKR, the molecular function of this gene are largely unclear both in vitro and in vivo, that motivating us to study this variant in more detail. So taking advantage of gene editing in PNPRA3 variant, we recently discovered that the risk variant PNPRA3 GG carrier spontaneously develops steatosis under normal condition. This is quite different from GCKR variant, which requires exogenous fatty acid for inducing pathogenic phenotypes. In addition, we performed a cohort of genotype-guided healthy volunteers recruited in Japan, and we found that among known genetic variants associated with NAFLD, only PNPRA3 associated with the history of fatty liver in past physical examinations. So these results are indicating that PNPRA3 variant may predispose to pre-symptomatic steatosis, but not to NASH itself. So to summarize in this story, we established unmasked liver organoid analysis, and using the gene editing, we uncovered the impact of GCKR variant on lipogenesis in insulin-resistant liver. Combining with the cohort analysis, it turns out that this variant has opposing effect with or without diabetic conditions. And as a preliminary data, PNPRA3, another risk variant for NAFLD, may predispose to pre-symptomatic steatosis. We believe that our organoid strategy presented in this today will offer mechanistic, diagnostic, and therapeutic toolbox toward precision medicine for fatty liver diseases. So thank you very much, and happy to take questions.
Thank you, Yosuke, for a great overview of your work, and I'm looking forward to seeing this progress in the future. We now have a time for a few questions from the audience. To ask a question, please put your question in the QA chat box, and I will read the question. From Ritika, incredible screening capacity. Thank you for the talk. Can you differentiate your 24 lines into organoids? Are there any line-to-line variations in the identity or ratio of cell type generated? How do you think this may influence the finding from pooled data?
Thank you for important questions. Yes, we are definitely taking care of the differentiation capacity of iPSC lines in this study. Actually, we modified the differentiation protocol in terms of the foie progenitor stage, so we can modulate the differentiation into mature liver organoid at this stage. So in this study, we can say differentiation capacity difference do not impact our screening results. But yeah, your question is very important. Thank you.
Okay, next question. Interesting talk. Have you had any plan to see how genetic variation affects the immune cells inside organoids?
I appreciate this question. Now we are tackling this kind of a challenge in our lab, but the problem is our organoid has a very few population of immune cell cell types in our organoid system. So currently, it might be a little bit difficult to test which kind of genetic variants impact the immune cell behavior, but we are very interested in. Thank you.
There are so many questions, but we have to move on to next speaker.
Okay.
Thank you again, Dr. Yoneyama, for your presentation. We encourage interested audience member to contact Yosuke to keep the conversation going. I will now hand it over to Ritika to introduce our next speaker.
Thank you so much, Yoshitaka, for handing over, and thank you for our speakers so far for fantastic talks. It is such a pleasure to be online and host this seminar for you today. My name is Ritika and I'm based in Australia, and I am excited to welcome the third and the last speaker of the session. But before we begin, I'd like to acknowledge the traditional owners of the land on who we dial from today, the Wurundjeri people of the Kulin Nations, and pay my respect to elders past and present. And I'm very excited to welcome our third and last speaker for this session on behalf of the Australasian Society for Stem Cell Research, AEST, Dr. Osvaldo Contreras. Osvaldo is currently a postdoc researcher at Victor Chang Cardiac Research Institute in Sydney, and will be speaking about their work on unraveling the effects of thymidine analog EDU on the cell cycle of human pluripotent stem cells. So Osvaldo, please go ahead and begin your presentation.
Hi, Ritika. Thank you for your introduction. I cannot start my video, says that.
See you yet? Okay.
Yeah. Perfect. Cool.
Awesome.
So everyone can see me now. Can you see my screen well?
Yep, it's looking great.
Perfect. Okay, thank you for your introduction, Ritika, and thank you to the organizers as well for the opportunity to present my work today. And to get it started, I'm going to give you first a glance of the importance of the cell cycle and highlight a few findings of our very recent work, less than, I'll say, a year of old. So the cell cycle is a fundamental process in cell division, playing a crucial role in various cellular functions, such as cell commitment, tissue formation and development. And it is also key in adult to maintain homeostasis. In this context, we want to understand its core phases as well as the regulatory mechanisms, because these regulations of the cell cycle can lead to congenital malformations and disease. The human cell cycle, just click that one, maybe the highlight to laser point. The human cell cycle consists of 4 major discrete phases, starting at G1, followed by S phase, or synthesis or DNA replication phase, then G2, and finally mitosis. And researchers have used different tools and strategies to model or identify the different phases and progression of the cell cycle, and this includes K67, phosphohistone-free, Aurora B, among others, as well as dual reporters such as Fuji, or in combination with DNA dyes to evaluate DNA content. Oops, something happened here. Perfect. Okay, I'm back. And remarkably, different detection methods have been used for evaluating DNA synthesis that happens in S phase of the cell cycle, including, for example, and they started with radiation detection of thymidine, followed by antibody detection of BIDU around the 80s, and more recently, the click chemistry-based assessment of EDU uptake. Since its discovery in 2008, EDU has been widely utilized for the analysis of DNA, replication, cell proliferation, and differentiation, and also for the accurate identification of the different phases of the cell cycle, as you can see in this flow cytometry plot, starting by G1, S phase, or G2M. It has also been used for high-resolution microscopy of DNA replication sites, as well as chromosomal labeling, as well as root plant growth. And notably, EDU has also been revealed or helped to reveal the proliferative nature of the stem cell gut niche, as well as neurogenesis, and ultimately, just recently, to understand DNA synthesis in postnatal hearts. However, despite its widespread adoption, prolonged EDU exposure associated with cytotoxicity. And nonetheless, or however, the underlying mechanisms of the cytotoxicity remain poorly understood. And on top of that, hundreds of studies have neglected the proposed EDU-mediated cytotoxicity. And I mean, to address this key point, some authors in this 2011 study recognized the need for improved thymidine analogs that could overcome the limitations of EDU. And as a result, they created or synthesized a novel non-toxic thymidine analog that is called FRA-EDU, as you can see in my slide, which is a second-generation thymidine analog. Notably, the use of FRA-EDU is still in its, I'll say, earlier stages, and there are only a limited numbers of publications using this analog, as you can observe here, only 5 up to date. We have recently studied using FRA-EDU, and we have validated, and we understand that the underusage of FRA-EDU is caused mainly because the lack of availability for kits to identify FRA-EDU. In our own investigations, we noticed that EDU exhibited considerable toxicity to human pluripotent stem cells. And by employing a strategy exactly the same, using the previously mentioned study, we observed that the IC50 toxicity of EDU was around the nanomolar range instead of the micromolar range that was previously described using immortalized cell lines, such as 3-3 fibroblasts. Further examination revealed that most cells die when labeled with EDU for only a 2-hour pulse, as you can see in this outline here. And those that remained after 48 hours exhibited chromosomal instability, including micronuclei formation. And I think these findings paved the way for further exploration. And to continue, what we did was basically to evaluate the effects of EDU on the proteome of iPSCs using untargeted proteomics. Our analysis revealed that EDU altered the levels of several proteins within iPSCs. And although I don't have time to give you details of each of these at the different time points, I want to highlight the following. Reactome pathway analysis shows that these protein alterations associated with several biological and cellular processes involving cell death, DNA damage and repair, and mitotic cell cycle regulation, which naturally led us to investigate the DNA damage response in pluripotent stem cells. Our initial investigations focused on the activation of the DNA damage response and sensors, and then signaling cascades of it. We observed that EDU at different time points induced ATM and ATR phosphorylation, as you can see here, indicating the presence of DNA damage and also the initiation of the DNA damage response. To further investigate the consequences of EDU-induced DNA damage, we examined the recruitment and activation of downstream effectors, such as BRCA1, gamma2A.X, and checkpoint kinase 2, which are proteins involved in DNA repair and cell cycle regulation. As you can observe here in this Western blot analysis, they were all found to be hyperphosphorylated soon after EDU exposure, indicating their involvement in the cellular response to EDU-induced DNA damage. P53 phosphorylation was only observed at 24 hours, indicating a delayed response to EDU, which suggests a secondary and delayed DNA damage response. Altogether, these results not only suggest that EDU could cause DNA damage, but also triggers cellular mechanisms to halt or to stop the cell cycle progression and initiate the DNA repair process. To evaluate the effect of EDU on cell cycle, and in this case, mitotic activity, we stained mitotic cells with phosphorylated histone H3 and observed that EDU impaired the mitotic entry of pluripotent cells time dependently, as seen in these percentages here. Based on these results, two possible explanations should be considered. The first one is that EDU blocks completely the progression of the cell cycle. And the second one could be that it actually delays the entry into G2 phase and then subsequently affecting mitosis. And to investigate both options, we examine how many mitotic cells, so you can observe here with the same marker, could be labeled with EDU at the different time points. Remarkably, all mitotic cells were labeled with EDU at the different time points, which implies that cells incorporating EDU in the S phase are indeed entering G2 and then mitosis. However, as we observed before, a decrease in the proportion of these cells indicate that EDU actually diminishes the rate or the speed at which cells enter mitosis. To gain a deeper understanding of the effect of EDU on cell cycle dynamics, we employed a fluorescent ubiquitination-based cell cycle indicator called FUCI. This innovative system utilizes cell cycle-dependent accumulation and degradation of two key proteins, CDT here in yellow, and germinin, in red, and germinin here in yellow. As you can observe in this schematic, CDT accumulates as cells progress throughout the G1 phase of the cell cycle and subsequently diminishes within the G1 to S phase transition. And on the contrary, germinin begins accumulating during the S phase and peaks in the G2 phase of the cell cycle to disappear in mitosis. Hence, by 2 minutes. Okay. Hence, by monitoring the fluorescent signals of these indicators, we can better understand the dynamics of the cell cycle in these cells. Confocal microscopy and semi-automated analysis revealed that a 4-hour EDU pulse followed by a 24-hour chase experiment led to a significant reduction in the proportion of cells expressing germinin and CDT1. And we have further confirmed these results using flow cytometry as well. Notably, we have recently performed live cell imaging of these cells to evaluate the human cell cycle one cell at a time, which is not possibly using traditional methods that only measure the average of cell timing or a population of cells. With this, we have determined the doubling time. We can also determine the G1 duration in iPSCs, the S to G2 duration, as well as the mitosis rate. And we are developing manifold training and AI-based single cell analysis, which is done in collaboration with some other groups, which I hope I can present in the present soon, Now I want to make a stop and I want to kind of highlight, well, what is the role of these two key markers, CDT1 and germinin, in human cell cycle? The delicate balance between these two proteins plays a critical role in preventing DNA re-replication, ensuring that replication occurs only once in each cell cycle. CDT1 in this schematic in blue is essential for DNA licensing by recruiting the hexameric MCM227 DNA helicase here in yellow and forming a pre-replicative complex at each DNA replication origin in the G1 phase of the cell cycle. The regulation of this process also involves germinin and other cofactors. Remarkably, DNA licensing, although perhaps unfamiliar to many of you, is a pivotal process that initiates DNA replication and ensures the progression of the cell cycle. We then measure the total or unstructured levels of MCM227, and we observe high levels across G1 or all the phases of the cell cycle. However, we made an intriguing discovery upon extracting the unbound fraction of this helicase in iPSCs using a high salt buffer extraction to eliminate the soluble non-chromatin associated protein. We found that iPSCs license the DNA in different replication origins very quickly. They do also have a very short G1 that corresponds to our live cell imaging. And therefore, iPSCs rapidly license the DNA replication origins. And to end, using our quantitative flow cytometry-based strategy that involved assessing the proportion of DNA-loaded MCM227 cells and categorizing these cells into 4 distinct groups, including high, medium, low, and negative, we obtained compelling results. Our findings show that EDU has a detrimental impact on NCM227 mediated DNA licensing dynamics, as you can see in this heat map plot. And the impairment of DNA licensing became evident after 6 hours after EDU treatment and continued to progress over time. And notably, EDU treated cells for 24 hours exhibited the most pronounced effects on DNA licensing. So in summary, we have discovered that EDU exposure triggers DNA damage response and replicative stress, leading to apoptosis. EDU treatment lengthens the S to G2 phases, impairing timely mitotic entry, disrupting therefore the cell cycle dynamics of pluripotent cells. However, some iPSCs are capable of overcoming the EDU-induced cell cycle defects, and they are capable of transitioning into the next cell phase of the next cell cycle and getting to G1. And all our findings, I think, are leading us into different perspectives. We want to continue investigating the human cell cycle, DNA replication and stress in genetic and acquired diseases using human advanced cell modeling and organoids as well. We are using multi-omics integrative approaches, and we have developed recently prolonged live-cell microscopy and analysis combined with flow cytometry, incorporating brand new wild-type iPSC cell lines that are capable of, because they are EGFP tag, to evaluate different cell cycle processes and dynamics. So having said that, I would like to finish by expressing my heartfelt gratitude to the Harvey Lab, our many collaborators that contributed to this project, the many facilities as well, and our funding sources and sponsors. And thank you everyone for listening to my talk today.
That was a beautiful talk, Osvaldo. Very, very exciting work. And I'm so keen to see what comes out, particularly around the AI training and the high throughput assessment and quantification of your single cell-based assays. And in true spirit of the ASSCR Rising Star winner from last year, Osvaldo, you're truly a shining light. We now have time for a few questions from the audience. So to ask a question, please just put it in the Q&A box and I will read it out. And maybe I can start. Have you tried to, with the Future Reporter, tried to, say, screen a dino-based or an organoid-based model of heart as well, as opposed to just the single cell at the minute?
Yes, we just started, and we're doing 3D tissue clearing using Wild Disco, a recently developed a clearing platform, as well as combined with light chain microscopy, but this is just nappies. And we got some organoids this week that we are clearing right now, actually. So that's the goal.
That is very, very exciting. And I have another question for you. Have you looked at the toxic effect of EDU in naive pluripotent stem cells? And Yosuke is guessing that you're using primed ones instead of naive ones?
Yeah, that's a great question indeed. We're using primed ones. We haven't, but it is something that we've been thinking of doing, especially using mouse naive versus prime ones. So it is a matter of discussion at the moment. And I think because you raised this point, we might have ended up doing it.
And we have one last question in the interest of time. This one's from Alex. When you design your experiments, how do you weigh the benefit of live imaging experiments with the large amount of data it generates versus just selecting several time points? And they said you had a fascinating talk.
Thank you for that, Alex. So I was, when we started this, we wondered the same. We generated 24 hours data sets, spaced by every 4 to 5 minutes. Which in total last week, using the Thunder microscope, we generated about 25 gigabytes of data in a single data set. And so we are not really struggling at the moment. I think it's fine. From the point of view of the data analysis is where we're kind of facing some issues on how we kind of reduce the quality, but when you reduce the quality, the segmentation also fails. So it is something we're dealing with. But thank you for raising that because it's actually an issue.
Thank you for answering those questions. And there's another comment that says incredible and inspirational, truly, Osvaldo, and to all our speakers there. So thank you again, Osvaldo, for taking the time out today to share with us a little bit about your work. We encourage any interested audience members to contact Osvaldo and to keep the conversation going. And this now sadly brings us to the end of our 3 talks for this session. I will invite the speakers and our facilitators to please turn on their cameras as we wrap up this session. Thank you to Alex, Yosuke, and Osvaldo for your excellent presentations today. I'd also like to thank everyone in the audience for their attention and all of the networks who are partnering on the speaker series to give a platform to rising stars in stem cell regenerative medicine research. And we hope you're feeling inspired because the future is indeed very bright. Also, a very special shout out to the ASSCR Victorian ECR Symposium that's currently happening. You can't see me, but I can see you. So wave at me because I can definitely see you. And we're looking very forward to seeing you at the next session for the Stem Cell Networks series. This will be happening in late September. And on behalf of the Stem Cell Network and the partner networks from Canada, Australia, Germany, Japan, Belgium, and Korea, thank you all for attending and we wish you well. See you later, everyone.
Bye.